today's lecture, we'll talk about immunolabeling, and this is a commonly used laboratory technique that allows for the visualization of antigens within a tissue sample. These practices are commonly used to identify different cancers and diseases, as well as allow medical professionals to appropriately plan treatments for their patients. Down at the bottom right, we see two side-by-side pictures of a tissue that has been immunolabeled with different labeling agents. In this case, we are using nanogold and then a control. The control is on the left, the nanogold is on the right. We can see this picture is comparing the strength of the two signals by how bright the antigens are shining. And we'll talk a little bit more about the different types of labeling agents that can be used for immunolabeling, as well as the different subsets of immunolabeling. Additionally, this presentation will discuss different types of immunolabeling, why it's used, and some advantages and disadvantages to the process. Okay, so the more academic definition of immunolabeling is that it's a process that detects and localizes antigen to a particular site within a cell, tissue, or organ. Now, there's two different subsets of immunolabeling, which we'll focus on. The first one being immunohistochemistry, and this is meant to label larger structures, and it's more common for locating antigens in tissue. So this is the one that we're going to be using a little more frequently for this presentation. However, we also have immunocytochemistry, which is meant more so to reveal information about a cell or its substructures. Since the overall immune response and antigens in general are a little bit bigger than cells, we're not going to focus too much on this. We'll focus mainly on immunohistochemistry. Now, this technology uses antibodies which are conjugated to labeling agents in order to locate these antigens. And the antibody that is the most commonly used for immunolabeling process is IgG. So focusing a little bit more on immunohistochemistry, we can break this process down into two different types of methods. We have the direct method and then we have the indirect method. So the direct method is when you have a single primary antibody binding to a labeling agent, so it's already bound. So let's say we have our tissue sample here. Of course, we're not drawing this to scale, but let's say we have a sample of tissue and then we have an antigen sitting right on top of that tissue. We're going to take an antibody, preferably IgG, that is already bound to a labeling agent. Now, this labeling agent is usually a fluorophore or a chromophore. And once this labeling agent binds to the antigen, right here, this is our antigen, we can shine light of a certain wavelength on it, and this labeling agent will light up and it will look very bright and neon. In this picture on the right over here, we can see this is what this neon marking is looking like. So this picture on the right shows a tissue sample that has been illuminated with either the direct method or the indirect method, which we'll see in a second. And it lights up after the immune response has been elicited and antibody has found antigen. And now the lab technician can go ahead and perform several other tests to see what kind of antigen this is. Now in the indirect method, we're not using one antibody, but we're using two. So once again, we're going to take our tissue sample over here. And this method is a little bit more special because you can use many different labeling agents and you can have different outcomes. So once again, here is our antigen on that tissue. We're going to bind a single antigen here, and then we're going to take another antigen. We're going to call it the secondary antigen and bind it to that primary antigen. So here is our primary antigen, and here is our secondary antigen. Now the secondary antigen is the one with the labeling agent on it. I'm going to put an L because it doesn't necessarily have to be a chromophore or a fluorophore, but it can also be a complex that once the immune response has been elicited, it can stain the surrounding tissue. So we can either have some sort of immunofluorescence like we did in this picture to the right, or we can essentially stain the tissue surrounding the antigen so that the lab technician can kind of understand a little bit better where this antigen is with respect to that tissue sample. So before going in depth about the direct and indirect methods and then giving you some examples, I think it's important to go over the basic antibody structure, what an antibody is, how it's been produced, and what it essentially does for immunolabeling. So if we remember, antibodies are produced by B cells, but during an immune response, which is why we're binding antibodies to antigens in a tissue sample for immunolabeling. We want that immune response to be created in order to activate that labeling agent. Now, antibodies are made up of two heavy chains, which are identical, and then two identical light chains. So each one of the heavy chains has one variable and several different constant regions, while the light chains have one variable and then one constant domain. So we see over here that this is the first heavy chain, this chain in red, which kind of wraps around the purple over here. And then we have the second one, which is the yellow heavy chain, and this wraps around and in front of that green chain. So the purple chain here and this green chain, those are known as the light chains. Now, the heavy chains over here are linked together by disulfide bonds, so they're non-covalently linked, and these ones up here are also linked. This region over here is where antigen binds to. So this is the AG binding region. And then we'll see, if you remember from that very brief description of the indirect method, this is where also antibody binds to, or IG. So when we have a primary and secondary antibody bonding together, this region over here, this variable region, or this Fab region, as we'll see in the next section, is where antibody or antigens bind to. This region over here, where the two heavy chains are linked together by the disulfide bonds, is known as the FC portion. So this is the FC region. Now, antibodies are usually in their classic Y shape, and each one has this sort of structure over here. This picture is a visual, this one on the left right here, of the different types of IGs. So types of IGs that are present in the body, and there's only 5 of them that we have discovered. But this is IgG, this is the one that is going to be used more so for immunolabeling purposes, but the other ones also play large roles in the body, just not necessarily for immunolabeling. This picture on the right over here shows a little bit more of a detailed antibody structure. So if this is an axis of symmetry and we fold the paper over, this part over here is going to be exactly the same and line up as this part over here. So we can see that this is the heavy chain, and it has our 3 constant domains, as I mentioned before, and then that one variable domain, whereas the light chains, which are pretty much colored in that darker green, they have one variable piece to them and then one constant region to them. Now that we have the basic knowledge and sort of that refresher of the antibody structure and what an antibody really does, where it binds to, we can go a little bit more in depth now between the direct and indirect methods of immunolabeling. So to start off, we have the direct method, and as I mentioned previously before, we're taking this primary antibody. So we only need one antibody right here. We're taking that and we're binding it to a labeling agent. So this labeling agent can either be a fluorophore or a chromophore. And the idea is that once this entire complex binds to an antigen on a tissue, we can take some sort of light source, we can shine it on the antigen, and it is going to irradiate with light. So this labeling agent is going to be irradiated. That was what that highlighted piece around the fluorophore was, and it's going to show light maybe around, maybe this region over here. But as you can see, it's not very generalized to just this one piece of antigen. Obviously, the closer we are to the antigen and the more strong the signal is, the better our signal is going to be. But with light, we know that it can irradiate outwards towards a much larger area and illuminate a larger area. It might even, illuminate this entire piece of tissue. We don't know. So that's why this method is not necessarily used as much as the indirect method is in laboratory settings. However, the direct method also has a much quicker processing time than the indirect, and it can limit the number of cross reactions that occur during these kinds of tests. But as I've said before, the direct immunolabeling method is not as sensitive as the indirect one, and it typically provides a less powerful signal. Additionally, it's more costly and less flexible with the labeling process because this process requires an abundant supply of the purified immunoglobulins, so an abundant supply of these guys over here that need to be pre-labeled and pre-tagged with either a fluorophore or chromophore. And with the direct method, we really can't switch out between fluorophore or chromophore. That's sort of the only type of labeling agent that we have there. And this can become very costly, and that's pretty much why we don't use that too much in a laboratory setting. Moving on to the more popular form of immunolabeling, the indirect method. This is the one that's used more so in labs for testing and staining and all sorts of things like that. But indirect immunolabeling happens when you have a single antigen and you bind that to a single antibody. We'll call it the primary antibody. And then you take another antibody or a third antibody or something like that with a labeling agent, whatever is the last in that chain, has the labeling agent, and you bind it to that initial antigen antibody complex, and some sort of reaction will occur. You can either shine that specific wavelength of light to induce immunofluorescence, like we saw in that neon colored tissue previously in a couple slides before this one, or you can have a complex. One of the major complexes used in laboratory testing is this ABC staining complex, or the avidin biotin complex, which I will talk about in a couple minutes. Now, there's many forms of this process, but essentially you can end up, as I said before, with either immunofluorescence or tissue staining. Now, the tissue staining is a little bit more precise, but this is one of the advantages of the indirect method. And we see here in this diagram, we have our primary antibody bound to the antigen. Once this occurs, we can call upon our secondary antibody with the labeling agent, or we can repeat this process, have another antibody come on here, and then have a tertiary or a fourth antibody, something like that, with a labeling agent. But for this case, we just have two. So the secondary antibody bears the labeling agent. Now, we can either shine light on this in order to induce immunofluorescence. You see this irradiation here with this highlight that surrounds it, or we can actually stain the tissue right here. So this is the stained tissue right here, this brown. As it turns out, with the indirect method specifically, we come across the issue of staining tissue that is not necessarily the target tissue. So there needs to be some sort of way to alleviate this and remedy this, so we can actually use blocking reagents. Now, blocking reagents come in three different types. We have the protein solutions, we have normal serum, and then we have protein-free commercial buffers. Now, if you're looking for something that lasts a little bit longer, I would suggest the protein-free commercial buffers. Normal serum is a biological agent here, and then the protein solutions, this could be some sort of bovine solution or something of that sort. But that has protein. It's not really too good for shelf life. This is probably the worst one for shelf life. But this one lasts pretty long. So this one over here is long lasting. Now, these essentially help prevent unnecessary staining of non-target tissue. So, as we saw with the immunofluorescence, light can pretty much irradiate an entire tissue sample, and that's not really what we're going for, which is why the direct method is not very much used in a laboratory setting. Now, these blocking agents have a very high binding affinity to the non-target reactive sites. Their reactive site is the area where antigen is not. And they actually compete with the antibodies used in lab, and they beat them, they outcompete them, these antibodies, and block anything surrounding the tissue, any antigens surrounding the tissue that doesn't necessarily need to be visualized for whatever tissue sample. So using blocking reagents is a very good way to do this. So again, I'm just going to draw a very simplified sample of tissue right here. And here we go. Here we have these lipid bilayers right here. Okay, so we have the antigen that we don't really need, and here is that pentagonal antigen that I've been using for our purposes. And here is another just plain antigen. Maybe this is in the gut or something like that, and there is some microbacteria that we do need. So these are the good ones, and this is the bad one we're trying to get rid of right here. So our lab antibody. This is this one, right? And it has the labeling agent. Our blocking reagent antibody is this one. Doesn't necessarily have anything on it, but it needs to bind to these good antigens. And this is non-reactive. It doesn't elicit any sort of immunological response, but it's essentially going to bind to these before our labeled antibody can get to it. So now the labeling antibody has no choice but to go right there onto the antigen in question, which is pretty much what we want it to do. It's going to bind, so these blocking reagents are essentially going to bind to the non-target epitopes. So this is the epitope right there to prevent our necessary antibodies, our lab-based antibodies, from binding to the non-target antigen. So moving on to one of the more common forms of indirect immunolabeling, it's known as this avidin biotin complex, or ABC staining. This is commonly used in the lab. It's very easy to buy this in multiple units commercially and essentially use it for immunolabeling tests. It involves two molecules, so we have avidin right here, and it's tetravalent. So let's just say that this is the tetravalent molecule. It has 4 different binding regions here. Okay, this is avidin, and it has a very, very strong binding affinity to biotin. Biotin is present in very trace amounts in the cells, and pretty much every cell in our body. It's known as vitamin H. People take this for healthy hair, skin, and nails, but this is vitamin H. So we can have up to 4 different molecules of biotin binding to this avidin here, and it is a very strong reaction. Now this reaction is a very important one because it calls upon the use of something known as horseradish peroxidase, or HRP. And HRP catalyzes a reaction that we'll see, which enables tissue staining. So we have a primary antigen that's first attached to the immunoglobulin, or vice versa, and then we have a secondary immunoglobulin that has this complex right here, this avidin biotin complex. Usually it's about 2 biotin molecules, not all 4 are hooked up over there. But then this attaches to the primary antigen. And then it calls upon HRP, which will catalyze a certain reaction. So the staining reaction is better seen here in this picture. We have our primary antibody right here, and it's attached to our antigen. And then we have a secondary antibody, which has this biotin avidin complex right here, and it's ready to go. Now this complex is going to call upon HRP, which, as mentioned previously, it's known as horseradish peroxidase. So it's going to call upon this HRP, and HRP is essentially a catalyst. So this is a catalyst, and it will catalyze a reaction between DAB, which is diaminobenzidine, and H2O2, which we know as hydrogen peroxide. So this is what's kind of going on here. We can take this a little bit downwards, and we can say this is our DAB molecule and our hydrogen peroxide molecule, both which are found in the body. But if not, again, we're taking this tissue sample out into the lab. We can add these reactants on here, and then we're going to take our HRP catalyst. Remember, it's not used up in reaction, it's just a catalyst. And it's going to give us something like carbon dioxide, and then it's going to give us a DAB precipitate. going to give us something, maybe not carbon dioxide, maybe something else, but it's going to release some sort of gas, and then the dab precipitate. So this is what the dab precipitate looks like. It's these little brown molecules here. Once the molecules reach the tissue in question, they are going to stain. And this is a little bit better, of course, if you use a blocking reagent, so we can pretty much have this entire thing stained here. Remember, our antigen is over here, so we're going to need this entire thing stained, but we don't want anything down here to be stained. That would not be good. So that's why, in addition to biotin, avidin staining, we're going to need our blocking reagents to help us out and localize this staining to where the antigen actually is right here. So this is good, but this is a little bit better. So if we want to put all of this into perspective and kind of realize why the indirect method is a little bit better for our laboratory purposes, as opposed to the direct method, which is a little bit more, I don't want to call it an archaic method, but it's not used as much as it was today as it was before. So for the direct method, some advantages of it are it has a quicker processing time and it mitigates cross reactions. We don't necessarily need blocking reagents for this. However, it is more expensive because we need an excess amount of purified antibodies that are already tagged with their labeling agents, and this can be hard to find commercially. But I will say that sometimes the direct method is used in lab, just not all the time, but it is useful when you have multiple antibodies from the same species existing on your tissue sample. Now, the indirect method is a little bit better. It's more sensitive than the direct method. Remember, if we're illuminating something with light, so light can irradiate further. Light can irradiate a little bit further than your antigen sample in question. So once again, if this is your tissue, a very poor drawing of a tissue, but if this is your sample, we'll make it look like this, some sort of very weird arbitrary shape for our antigen, right? And we have our primary antibody with a fluorophore, and we shine the light on it. This is not only going to illuminate all of this region over here, but it could also illuminate all of this region, which is a much broader, less defined area, and it's not really useful for pinpointing the exact location of antigen, but the indirect method is a little bit better with this in that it's more sensitive and it pretty much details your exact location. Not very exact, but much more exact than the direct method's immunofluorescence would ever be. Additionally, it's easy to change the signal color. So as mentioned previously with that avidin-biotin complex staining, we can have brown, but we can switch out the avidin-biotin complex for some other complex that maybe produces a different color, maybe a red color or a purple color or something like that. And this is very easy to derive and obtain commercially. So easy to purchase for lab. One disadvantage, you could call it a disadvantage, but it helps with the overall process, is that you're going to need to use a blocking reagent if you're using the indirect method. And this is because, as we've seen before, this method doesn't really mitigate cross reactions. If anything, it gives light to more cross reactions, because the more antibodies you're putting on a surrounding tissue sample, the more apt beneficial or non-target antigen epitopes can be to these introductions of new antibodies. So we need to use blocking reagents. That could get expensive, it could get a little more time consuming, but it's just something it's an extra step to take into account for this overall process. Now, despite the method of immunolabeling that you use, both of these, the direct and indirect, have helped physicians and laboratory researchers in the long run basically pinpoint what kind of antigen you have, where it is, how it can affect you, and then further help treatment plans get a little bit better for patients and extend lifespans and things like that. So overall, immunolabeling has become a very important invention for medicine to this date, and it's still being worked on today. We're seeing, as we saw in that previous title page slide, nano gold. So we have different very nano structures of gold, different colloidal golds that we can use as immunolabeling agents that are working and can be picked up through different imaging techniques like electron microscopy and things like that. Either way, immunolabeling is critically important to practical immunological techniques, making it a crucial and very exciting area of research for the future.